Now students, we are going to discuss that what is hematocrit value. So if blood is collected in a tube, that is hematocrit tube, along with an anticoagulant which is already present in your lab, you can calculate the packed cell volume, and this packed cell volume actually represents the hematocrit value. Now we will discuss in detail that what is packed cell volume, what is hematocrit value. Now take a tube, that is hematocrit tube, collect blood in it. Now add anticoagulant in it. Now place this tube in a machine, that is centrifuge machine. Set the machine at the speed of 3000 revolutions per minute, that is 3000 RPM. RPM is abbreviation for revolutions per minute. Now run the machine for 30 minutes. After 30 minutes, when you will take the tube out of the machine, you can see that the red blood cells have settled down. So, however, the plasma is at the top of the tube, and there is a buffy material in between this plasma and red blood cell. So, the actual volume of the plasma in the tube can be 55%, and the volume of the red blood cell is from 40 to 45%, and the volume of this buffy material is 5%. So, according to the book Essentials of Medical Physiology, as he is just discussing hematocrit value, he just indicated that plasma is 55% and your red blood cell is 45%. But according to certain researches, plasma is 55%, your blood that is at the bottom is 40 or 41%, while the volume that is in between this plasma and the red blood cell is 4 to 5%. Now, the volume of red blood cell that is at the bottom is actually representing packed cell volume, and this is expressed in the unit of percentage, and this percentage is actually the value, that is hematocrit value. How you can calculate hematocrit value? Let's take an example that you have a blood column. This is the tube. It has marking in millimeter, that is mm, and the blood volume in total in this tube is 50 mm. However, for example, the red blood cells in this tube is 20 mm. So now how you can calculate the hematocrit value in percentages? You will divide this packed cell volume, that is 20, with the blood column measure, that is 50 mm. Now after dividing, you get a value that is 0.4. You will multiply this value with 100, and the resultant will be in percentages, that is 40%. Now this 40%, that is this 40%, which is representing the packed cell volume, which is representing the red blood cells, is actually the hematocrit value. So in between this plasma and this red blood cells, we have white buffy material, which is actually the red blood cells and the platelets. So this was all about the hematocrit value.